At the heart of every living cell lies a molecular instruction manual. In this unit, we explore the central dogma of molecular biology, the directional flow of genetic information from DNA to RNA, and finally into the proteins that build and operate your cells. This is not just a memory exercise in biochemistry. It is the absolute foundation for everything you will study in genetics, cell division, and biotechnology. Cambridge examiners use this unit to ruthlessly test your precision. We are going to move beyond simple diagrams and master exactly how these molecules interact at a chemical level.
Before we can read the genetic code, we must understand the molecular vault that stores it. You will master the architecture of nucleotides, focusing on how they link via phosphodiester bonds to form a sugar-phosphate backbone. We will compare how DNA forms a highly stable anti-parallel double helix to protect your genetic blueprint, while RNA uses subtle differences, like a ribose sugar and the base uracil, to function as a temporary mobile messenger. Getting these structural differences clear early on is an easy way to secure marks in Paper 1.
Every time a cell divides, it must copy its entire genome flawlessly. You will learn the exact chronological sequence of DNA replication during the S phase of the cell cycle. We will track how helicase unzips the helix and how DNA polymerase builds the new strand. Crucially, you will understand the strict 5' to 3' directionality that governs this process. We will also analyze the classic Michelson and Stahl centrifuge experiment using heavy nitrogen isotopes, which provided the definitive proof that replication is semi-conservative.
So, how do sequences of just 4 nucleotide bases translate into sequences of 20 different amino acids? You will learn the rules of the genetic code. We will see how it is read in triplets, or codons. You will understand exactly what it means for the code to be non-overlapping, why it is practically universal across all living organisms, and why its degenerate nature is a brilliant evolutionary defense against harmful mutations. This is the biological dictionary you need to decode any gene.
Finally, we bring the entire narrative together in protein synthesis. It happens in 2 major stages. First is transcription, where the permanent DNA code is transcribed into a temporary messenger RNA copy inside the nucleus. Then comes translation, shown here. The mRNA travels to a ribosome in the cytoplasm. Transfer RNA molecules, those folded structures you see, act as the physical translators. They read the mRNA codons and deliver the correct amino acids one by one, building a specific, functional polypeptide chain. The sequence of events here frequently forms an 8 to 12 mark question in Paper 2, and you will learn to describe it flawlessly.
This unit is a sequential journey, from the static structure of a nucleotide to the dynamic copying of a chromosome to the active expression of a gene. Master these steps and the rest of the biology syllabus will click into place.